if you remember during mitosis, I told you that there is actually, before any cell division happen, we have something that we call the cell cycle. The cell cycle involves interphase, the mitosis, and then cytokinesis. And I told you before any cell division happen, there will be interphase. And it's as good as before you come take any exam, you should have studied for this. And you are going to come take the exam for just an hour. Now, the same way it is how mitosis is. Mitosis is the preparation stage. That is when you are preparing for the cell division. Now, when the cell gets to division phase, it is already well prepared. And I told you mitosis have three stages, the G1, S phase, and the G2. G1 means gap phase or growth phase, and G2 means gap phase or growth phase 2. Now, so there's going to be one interphase, and then the cell division prophase, metaphase, anaphase, telophase happens with cytokinesis. But in meiosis, it is different. The difference is this. There's going to be one interphase, just like we have in mitosis. There's going to be one interphase. The cell will grow its proteins, grow its uh, organelles. It will duplicate its DNA and ready for division. But when it's ready for division, meiosis happen two times. So there's going to be meiosis 1 and meiosis 2. And if there's meiosis 1, that means in meiosis, there's going to be prophase 1, metaphase 1, anaphase 1, telophase 1. Then after that, there would not be interphase. It's just one interphase, then meiosis 1 and meiosis 2. So after meiosis 1, there's going to be meiosis 2, which is prophase 1, prophase 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. So in quotes, the difference between meiosis and mitosis is that in mitosis, there's one interphase and one cell division. In meiosis, there's one interphase and two cell divisions. So like mitosis, meiosis is preceded by replication of chromosome, which happen in the S phase of interphase. Chromosome duplicate before meiosis will happen, of course. The resulting sister chromatids are closely associated with their length. Along their length, this is called sister chromatid cohesion. The chromatids are sorted. Oh, great. Great. So someone just shared with me that they call it P something P on mat, right? So I use the word IPMAT. IPMAT is interphase, prophase, metaphase, anaphase, and telophase. IPMAT or PMAT. So P on mat on the mat. That is PMAT. P on the mat. Thank you. So the chromatids are sorted into 4 haploid daughter cells. Like I told you, there are 4 hard haploid daughter cells that are not identical as the parent. So we call them 4 haploid non-identical, and none of the 4 are the same as well. I'm going to show it to you. These images make it look like they are the same, but I'm going to show it to you at the end, and you're going to see the beauty of mitosis. Of meiosis. So invariably, what happened in meiosis is what you can see here. Please take note of this with me. I will explain some things now that may look like mystery, but you're going to understand it and it will make sense. Now, this is the chromosome that you have. Think about it as I'm talking about you now. This is the chromosome that you have from your parents. All right. So you have gotten the blue from your father. You got the red from your mother. Now, before these cells will go ahead into meiosis, they are going to go through what we call mitosis. And in mitosis, it means that in interphase rather, they will grow and duplicate. And so now they have duplicated. So this cell has already undergone interphase. Now the next thing that will happen is meiosis 1. When meiosis 1 begins, you see this comes here, this comes here. At the end of meiosis 1, we have this here, we have this here. Now let me ask you an interesting question. This here is a set of chromosomes, right? Yes. What makes it a set? There is a red and there's a blue, meaning that there's some chromosome father and from mother. That makes it a set. A set does not mean two. Two that are the same is not a set. If you have two right shoes, do you have a shoe to wear out? If you have two right shoes, do you actually have a shoe? No, you do, you don't have a shoe. You only have two pieces of some materials. If you have two left shoes, do you have a shoe? No. So you have a textbook, and so I have my daughter's textbook here. You have this textbook, and this textbook is, I have on this book now. Let me show you something very interesting. On this book, I have about 760 pages. If I say, okay, I want you to read this book, and then you, what is half of 760? The half of 760 is going to be 350, 360, 380. So the half of 760 is 380. So if I give you this book now, and I give you 380 pages, from page 1 to page 380. Sorry, half of 760 is not 380. 769 is 3. Can you imagine? Sorry, that's my phone. If you have 760, 700 is, if you divide into 2, is 350, right? So if you divide the 60 into 2, that will be 30, 30. So 350 and 30. Oh, that's 380. I'm correct. Sorry. So if you have 380 and I gave you page 1 to 380, if you have page 1 to 380 and I ask you to read that, and you read it 2 times, or I made 2 copies of it, you have read the whole pages of the book in terms of number, but have you read the whole book? Reading half of this book twice, did you get the content of a whole book there? No. The same thing with a shoe. If you have 2 right shoes, you don't have a shoe. So what happened here is this. Look at this. I'm going to show this. If you understand this, then that's actually the content. So this here is, this is the from the father. Now this became duplicated, and now you have 2 of these. Now then at the end of meiosis 1, you have this one here now. Then the same thing for the mother, duplicated, and now you have 2 of it here. You now have the same 2 here. Now think about it. In this cell, this parent cell, this germ cell, you have a complete set of chromosomes there, one from father and one from mother, right and left. That's why it's complete set. The father is 2. Here, it's not a complete set. Now, we are going to say this cell is 2n, and we say it is diploid. So we say this cell is diploid. I'm sorry, the way I write is because I use my mouse. Now you say this cell is diploid. But when you come here, now, do you think we can say this cell is diploid? No, because it's not a complete set. This here is just double of the mother. There's no child that have chromosome from the mother alone. There's no child that have chromosome from the father alone. A complete set of chromosome is half of the father and half of the mother. So now when a cell have just double of one parent, it is not complete. So even though in terms of number, it is complete, just like someone that have 2 right shoes, of course, you just need 2 pair of shoes. But a pair of shoes is right and left. So having 2 right shoes does not give you a complete shoe, even though in terms of number it's complete, but in terms of usage, it is not. So in terms of number, you have two sister chromatids here. Like I told you, sister chromatids means half of a chromosome, half of a chromosome. You have two sister chromatids here, but of course, they are the same from the same person. That cannot contribute to a new individual. A new individual should have half of each parent. So having this is not a diploid again. At this point, we consider this cell as, in quotes, N and another N. So actually, this cell at this point is haploid, not diploid. So we say this cell at this point, it is deep haploid. The reason it is haploid is simply because there are two copies of the same chromosome. So it doesn't have a full set, it only has two of the same thing. Does it make sense to you? Okay, great. So we say that the cells are haploid right from meiosis 1 because meiosis 1 just double one of each chromosome into the cell. It is not the complete set, like having two pairs of shoes, right and left. So in quotes, you have right here, you have left here. You are not going to, that the fact that you have two of them does not mean you have complete set. All right. Now, at the end of the day, meiosis 2 is going to happen, and then the cell will now split into, so each of these N will now split. You have N here again, you have N here, you have N here, you have N here. Now we have four diploid, different, like non-identical haploid cells now. Let's go. And that is actually what happened in meiosis. That's actually all. Every other thing we are looking at, we're just looking at the flesh to see how a lot of other things happen there. So meiosis takes place in two consecutive cell division. We call it meiosis 1 and meiosis 2. The two cell division result in four daughter cells like you see, and I told you the four daughter cells are haploid and they are non-identical. Rather than the two daughter cells you see in mitosis, each daughter cell has only half. You see that? It has only half of many chromosomes as the parent cells. Just half, haploid. Division in meiosis 1 occur in four phases. We have prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. Then in meiosis 2, so I'll talk about this later. In meiosis 2, again, it's going to be, so right after that, you're going to have meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. Now let's go and look at each of the descriptions of the stages. Very interesting. Now, in prophase 1, just like what happened in prophase, in metaphase, but there is a little more here. Significant thing that happened here is what determines differences in genetic diversity. That is the bedrock of the reason why the chromosomes are not the same, while the cells are different. You know, I told you the genetic composition of the cells are different. The reason they are different is actually because of what happens here in prophase 1. So prophase 1 is key. So in prophase 1, each chromosome pairs with its homologous and crossing over occur. So key thing that you may not want to forget that happened in prophase 1, we call it crossing over. Let me cross it over. Crossing over. That is what happened in prophase 1. The point where the crossing over occur, we call it chiasmata. I'm going to show the images to you. But crossing over happened in prophase 1. Now, what we call crossing over leads to, this is actually what happened in crossing over. Let me show this to you, even though I'm still going to show you the images. So this is a chromosome. Then let me use another color to show another one. And this is another chromosome. So in meiosis 1, they are going to cross on each other like that. So this is what we call crossing over, this point where they cross over. And the location of the crossing over, the location where the crossing over actually happened is what we call chiasmata. Now, what do you think is the essence of this crossing over? Let me just do it right here and show it to you so that you have a mental picture of it, so that even when we are doing it, there is You already know what it is. At the end of the day, when they will separate, of course, they will later separate. When they separate in anaphase, what's going to happen? This one, let me number them, and then you can see what I'm talking about. So let's say this is chromosome 1, this is 2, this is 3, and this is 4. Now, at the end of the day, you will see chromosome 1 is going to be only purple. Chromosome 2, of course, is going to be, oh no, I mixed it up. Let me start it again. Chromosome 1, you see 1, it's crossed over with 4. So chromosome 1 will, by the time you remove it, it will have a little portion of 4 on itself. This is the best I can do it with this mouse. So it will have a little portion of 4 on itself. So some part of 4 will be there. And we call that recombination. Because when they cross over, then they will recombine with each other. So we call it recombination. Now, number 2 did not cross with any of them. So it's going to be just purple. Number 4 also crossed with 1. So it's going to be purple and then blue. Now, number 3 is going to be blue alone. So if you look at this, you will notice that this is number 1, this is number 2, this is number 3. No, this is the ideal number 3 here, and this is number 4. So if you look at this, you will see that all of the 4 are actually different genetically. Not just that they are haploid now, they are all different because of the crossing over that happened in Prophase 1. So Prophase 1 does like a major path in the differences or variation that we have. So that's why you have, like now I have 3 beautiful girls, and none of them have the same face. None of them have, when you see them, you can tell that these are different. They have different traits. The shape of their nose are different. The a lot of things are different in them. Their set of teeth are different, all because there is the fact that they received a trait from me, they received a gene from me, does not mean they are going to have that gene in its like a copy and paste. No, even when you say, oh, this person looks alike like the parent, but by the time you keep checking each of the traits, you see that okay, she has a trait like the mother, she has the mother's face, but there's still difference because they are these are going to produce different cells in essence. Let's go. So in metaphase 1, you remember when we talked about how chromosomes the chromatin will condense and become visible as chromosome in chapter 12. So all of those also happen in prophase, all right? So the chromosome is condensed, nuclear envelope is beginning to disintegrate now so that the chromosome can get out, spindle fiber forms, which are the things that happen in prophase 1. So you may want to go back to chapter 12 to watch the video again if you can't lay hold of those things. So in metaphase, so that's why I'm just going to run through these things because the content of the chart of the stages are the same. It's just having little differences because now we are talking about different kind of chromosome. The chromosome in interphase has now become haploid chromosome in meiosis 1, then they are still going to be haploid in meiosis 2. So if you see a question that is asking you, what is the genetic composition of chromosomes in meiosis 1? Of course, is haploid. So in metaphase 1, what characterizes metaphase? We just know that chromosomes are aligned at the metaphase plate, at the equator, at the middle of the cell. In metaphase 1, the pair of homologous. Now, homologous now, we are not saying pair of chromosomes. We are saying pair of homologous because it's a pair of this. They've not at metaphase 1, they didn't separate yet. They are going to separate at anaphase. You remember that. So, but in meiosis, in metaphase 1, what has happened in prophase is that the chromosome has become coupled together, they join together. They still remain joined together. Now, at the point when they are joined together, how many sister chromatids do we have here? We have 1, 2, 3, 4. So at that point, we call this joined together. We say homologous chromosome because they are joined together, and we also call it tetrad. And the word tetrad is because there are 4 sister chromatids there. So in metaphase 1, pair of homologous line up. So they are going to line up just like the way they are at the metaphase plate, with one chromosome facing each pole. So they do go like this. Microtubules from one pole are attached to the kinetochore of one chromosome of each pair. The microtubules from the other pole are attached to the kinetochore of the other. So you just go that way. Let's go. In anaphase 1, they are now going to separate. But look at it. What is going to separate? The pair of homologous chromosome which was together will now separate. When the pair of homologous chromosomes separate, what's going to happen? Because homologous chromosome means there are 2 chromosomes. So what's going to happen now? One set from each pair will go to each side, opposite side, just like what you see happen in mitosis. But now we are not talking about chromatid, because now we have double. So it's homologous chromosome, then it's chromosomes separating now. Does that make sense? Is one chromosome going to different side, because we have 2 sets and one of each set is going to different side, not half going to different side. It's a whole one set, one of each set is now going to different side. Now sister chromatids remain attached. You see that? We are not talking about sister chromatids separating yet. They attach to the centromere and move as a unit towards the pole. See what's going to happen now. Now, in telophase, in the beginning of telophase 1, each half of the cell has a haploid set of duplicated chromosomes. Now, you see, we say haploid set of duplicated is like 2 sets of right shoes, or 2 sets of half of a book is still half of a book. Each chromosome still consists of 2 sister chromatids. You see that? Now, cytokinesis occur simultaneously, forming 2 haploid daughter cells. Now, these daughter cells, they are haploid. In animal cell, a cleavage furrow forms in plant cell plate form, just like we talked about. Now, no chromosome replication occur between meiosis 1 and meiosis 2. Now, let's go and look at what is going to happen. This is exactly what I've just shown you. Here, everything that we say happen in prophase normally is going to happen. Nuclear envelope begins to break, chromosome condenses, spindle fiber forms. Those are the regular things that is going to happen. Now, but then additionally, crossing over is going to happen, which is what you see here. And the crossing over will also lead to recombination. So this is what you see that has happened here. Crossing over has happened. The point where crossing over happened, we call it chiasmata or chiasmata. So you see, that is crossing over happen. Crossing over happened there. Crossing over happens. You see that? Crossing over has happened. Now, so we call them. Now, this is homologous chromosome. This is homologous chromosome. So there are 2 chromosomes here now. And if you have 2 chromosomes, like I told you, this and this, they are sister chromatids because they belong to the chromosome of the father. And this also will be sister chromatids. They belong to chromosome of the mother. So sister chromatids is 1 strand. So that means in just this one alone, now we have 4 sister chromatids, and that's why we call it tetrad. So this is called tetrad because there are 4 sister chromatids there. All right. Now, but when you come to metaphase, see what happened. They attach to the middle by the centromere. And what is that? You see, by the metaphase plate, the chromatin spindle fiber then attached to each of them. You see that? Spindle fiber attached to one of each, attached to one of each, attached to one of each, like that. Then insist at anaphase 1, the chromosome here, these are homologous chromosome here, homologous chromosome. In meta-anaphase 1, chromosomes, homologous chromosome now separate, and then chromosome move towards together. But each chromosome are still together with sister chromatids. Does that make sense? Now, that is the difference in meiosis and mitosis. In meiosis 1, you'll be talking about homologous chromosome separating. That's the difference in meiosis 1 and in meiosis 1 and mitosis. Now, let me tell you something very interesting now. We are going to meiosis 2 now. Meiosis 2 is about the same thing as mitosis. Every of the terminologies are the same. In meiosis 1, we're looking at homologous chromosome. But now look at it. At the end of meiosis 1, sister chromatid is what is now here. So it is this, chromosome is there, connected together. The sister chromatids connected together to form a chromosome, which is actually what is now going to happen in mitosis. So in mitosis, we say that sister chromatids separate. That's what is going to happen here. So in meiosis 2 and mitosis, they are the same thing. So prophase 2, metaphase 2, and aphase 2, there is not going to be anything special happening in prophase 2 again. In prophase 2, spindle fiber forms, just like we talked about. In late prophase, chromosome again will condense and they move towards each other like that, moving towards the metaphase. Now, don't forget we talk about prometaphase in mitosis. It's still the same thing. There's no need for prometaphase as a major stage to note it again, but it's the same. Now, in metaphase 2, the sister chromatids are arranged at the metaphase plate. You see that? The sister chromatids, each of them making chromosome in coats, right? We call it sister chromatids. Now, you see, because of the crossing over in meiosis 1, the sister chromatids of each chromosome are no longer genetically identical. You can see that they are no longer identical. They are, and you can see that here. You see, they are all now different, right? Now, the kinetochore of sister chromatids attached to microtubules extending from from opposite pole. In anaphase 2, see what's going to happen. The sister chromatids are now going to separate, which is the same terminology we're using in mitosis, right? The sister chromatid of each chromosome now move as two newly individual chromosomes towards the pole. In telophase 2, the chromosome arrives at the pole, and then the nuclear envelope begins to form back. The chromosome begins to start to separate so that they are no longer condensed. So they decondense. All right. Then cytokinesis separate the cytoplasm. At the end of meiosis 2 now, there are four daughter cells, each with haploid set of unreplicated chromosomes, and each daughter cell is genetically distinct. Those are the keywords that you want to not forget. So I just say that we have four haploid non-identical daughter cells. So I'll put that here the way I quote it so that you don't forget that. So I say we have four four haploid non-identical daughter cells. All right, that's what we say when we get to that stage. So that's what we call the product of meiosis, is four non-identical daughter cells, whereas mitosis is two identical diploid cells. So this is what you have. You have these stages here, just a review. You have prophase 2, what you have the homologous chromosomes. We have the chromosomes now that has been recombinated. So we call it recombinant chromosome. It has been recombined. They are going to align at the metaphase plate. You see, they are all now different. They have fragment of each other. Then sister chromatids will separate. Then now they are now at the poles, and then you are going to now have different cells forming, and that is what you see in meiosis 2. Sorry. All right. I will let you see this.
Eggs and sperm. Process of meiosis. Here, by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored. Most of a person's cells are diploid, two sets of chromosomes. Cells here in red. Set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These map pairs are called chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone protein. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
All right. So that is what we see in meiosis, and I'll quickly wrap it up now in a few minutes. Um, then I'll take questions if you have any. Crossing over and synapses during prophase 1, we just want to talk a little about it now. In prophase 1, after interphase, the sister chromatids are held together by protein called cohesion, and the non-sister chromatids um, are broken at precisely matching points. All right, so the so
Thank you,
now
Professor.
a zipper-like structure called synaptonemal complex holds the homologous chromosome together. So where we say the crossing over happened, I told you that the point where the crossover, so this is actually what we are talking about. The point where the crossover, we say that's the point where the synapse, and we call it chiasmata. Now, the location is what we call chiasmata. The location is called chiasmata. We're saying join them together, and we call the complex formed synaptonema complex. So when you see you see a zipper-like structure called synaptonema complex hold the homologous chromosome tightly. So the structure there is called synaptonema complex. The location, chiasmata, the protein that holds them together is the coenzyme that holds the chromosomes together. Now, mitosis conserves the number of chromosome sets, meaning that if 46 is going to give rise to 46, but meiosis reduces it. And so mitosis, I call mitosis nuclear, I just going to type it here. I call mitosis nuclear multiplication because invariably it is the nucleus that is multiplying in mitosis. So I call it nuclear multiplication, whereas meiosis, I call it nuclear reduction. It reduces the nucleus. All right. Now, three events that are unique to meiosis is the synapsis and crossing over, which happen in most prophase 1. Homologous chromosome physically connect and exchange genetic material, which will lead to recombination. Now, alignment of homologous pair at the metaphase plate, because homologous pair is aligning at metaphase plate and not chromosome, not sister chromatids. So that is key to meiosis as well. And of course, this is in prophase 1, this is in metaphase. Then separation of homologous in anaphase 1. Those are unique to meiosis 1. Now, I told you this crossing over is going to lead to recombination. And recombination is the reason the chromosomes recombination is the reason the sister chromatids, is the reason the daughter cells are different, right? Let's go. All right. So these are the same image we've reviewed. So I will just go. Let me see if there's anything else I still want to touch before we go. Okay, I've talked about this. Now, so the behavior of chromosome during meiosis and fertilization is responsible for most of the variation that arise in each generation. Three mechanisms contribute to genetic variation. So look at it. Independent assortment of chromosome. Number two, crossing over. Number three, random fertilization. Independent assortment simply means that chromosome separates without assortment is to separate, to sort. Independent means that it doesn't have to depend on any factor. It doesn't mean that this is the chromosome that have to segregate at this point. This is the chromosome that have to be given to this child. It is independent assortment. And that's what you see when you come here, for instance, now. When you come here, the assortment of chromosome in, let me go to metaphase. Okay, good. When you come here, the arrangement of this chromosome in metaphase 1, 2, or metaphase 1 does not have any other. It can happen anyhow. So this arrangement can happen like it can be this flipping and you have this on the right side and this on the other side. This can be on this side, this can be on this side. This can be on this side, this can be on this side. So it can be anyway. There's no order for this assortment. This sorting, this arrangement can happen independently of each other. You can flip it. There's no order that says it has to arrange this way. It can arrange the other way. And that means it can separate anyway. All right. So that's what we call independent assortment, which happen particularly in anaphase 1 and majorly. So that happens, then crossing over, of course, which will lead to recombination, and then there's random fertilization. All right. So these are explanation of the things I've talked about. Those talk about recombinant DNA. All right. So I believe, yeah, I'm done now.